15 discount tickets that he has, cash only, $25. But there's a lot of fun things that come along with that as well. So if you'd like to buy a ticket, Nathan's your guy. So we all came together, believe it or not, through one of our science cheerleaders who was not able to make it, who is in Houston. She used to cheer for the Texans, and she's an engineer at NASA. And she thought, you know, I've done enough of these science cheerleader appearances where we go out and we, you know, try to challenge stereotypes, and we get kids involved in fun science cheers, and we point them to different activities they can do. But the brain power among the science cheerleaders is enough to warrant some really, real good research. So it was really her idea to do something that taps the intelligence of the science cheerleaders. And she linked up with somebody that she's working with at NASA, who kind of brought to our attention, why aren't you doing something on the International Space Station? NASA's really looking for opportunities for people to do research up there. They built it, somebody should use it. So we started looking around for those types of opportunities. About the same time, I was meeting up occasionally with their professor, Jonathan Eisen from UC Davis, who I remember saying at some point, and it could have been the conference that David organized, it would be so much fun to have like mass participation with people collecting microbes that we can analyze and make more sense of. And so immediately I thought, like, mass participation might be San Antonio Spurs game or an NFL or an NBA game, and for that we have access to be able to do stuff like that. So some of our researchers who are not here today would be Summer from the Houston Texans, who works at NASA, Mark Severance, who works at NASA as well, he used to run their education programs, Wendy Brown, who is getting her PhD at UC Davis, and of course Jonathan Eisen, who's out, I guess he just got back from Costa Rica or something, right? But the rest of our team is here, and David will introduce them as he talks about the scientific merit of our project. Thank you.
So my name is David Coyle. I'm a project scientist at UC Davis. And I just wanted to briefly talk about the groups who are involved in this project. UC Davis, obviously, Darlene's mentioned some of the rest. NanoRacks and Space Florida are two different organizations that are involved in various aspects of sending projects, scientific research projects, to the ISS. And they held a competition to promote their business model, which is that people bring them experiments and they take care of all the logistics and paperwork to send it to the space station. And they offered, I think, 6 free experiments, and people could apply. And so Darlene and Jonathan got together and came up with this idea and applied, and we won one of those slots. So it's awesome because sending stuff to space is really crazy expensive, and we don't have to pay for it. So NanoRacks and Space Florida are paying for it, and then our time is being paid by the Alfred P. Sloan Foundation, which is a private foundation that's interested in research, and I'll talk more about that in the MicrobeNet project in a minute. So just a little background on where myself and Dr. Jenna Lang and Russell Netchus work. We work in a lab at UC Davis that looks at the ecology and evolution of microbial communities. We look at microbial genomics and phylogenetics. We also do a lot of metagenomics, which is where you just go into the environment and you grab a teaspoon of soil and you sequence all the DNA in that teaspoon of soil and figure out what was there and what's it doing. And as a result of this, we also do a lot of bioinformatics, which is sort of the computational assessment of all this sequence data that these other things generate. So we develop a lot of in-house software and tools to analyze the kind of data that our experiments generate. So that's just a little background on us. The project that I work on is this called the Microbiology of the Built Environment Network. This is a project funded by the Alfred P. Sloan Foundation to study microbes in the built environment. And basically the Sloan Foundation a number of years ago decided they wanted to create a new field. They thought not enough people were studying microbes in buildings and indoor environments. We know more about microbes that live in deep sea vents in the ocean than we do about what lives in an office building. They wanted to fix this, so they threw many millions of dollars at this problem, funding different labs to do different aspects of research. And then they brought us on board to do communication and collaboration, to make sure that all the people studying this new field talk to each other, to do public outreach, communication, those sorts of things. So that's the MicrobeNet project that's paying for our part of this Project Mercury. So what is Project Mercury? So there are 3 aspects of the project, which I'm just going to mention here, and then I'll talk about it in a little more detail. The first is studying the microbiome of the International Space Station. So we don't really know what's living on the International Space Station. And so that's the first question that we want to answer. We're also doing a variety of these events, like the Spurs game tomorrow. We're going to be taking samples from people and comparing those to the International Space Station. I'll talk more about that later. And then the last is this microbial playoffs in space, which is a microbial competition we're going to have on the space station. And if you're curious, Project Mercury stands for Microbial Ecology Research, combining citizen and university researchers on ISS. It's a little bit tortured, but it sounds cool. So the first aspect of this project is the microbiome of the ISS. So there are no published studies of the ISS microbiome. I mean, it's entirely probable that NASA has done this kind of work and it just died in a hole somewhere. So we want to actually figure out what's living on the space station and communicate that. There's been some work where people have done culture-based work, where they go and take swabs and they grow it up on plates. But that's all very focused on pathogens. You know, they're concerned about what sort of scary things might live on the space station. That's justifiable, but there's been no attempt to look at the ecology, you know, the sort of broad spectrum of things that might be up there. So the astronauts, as part of this project, the astronauts are going to swab a variety of surfaces on the space station, send those back to us, and we will analyze them and figure out what's living there. The second part of this is the swabbing stadiums and space meetups, and this is really the citizen science part of the project. This is engaging the public on a large scale. What we're going to have people do is we're going to sample cell phones and shoes at sporting events around the country. And the reason we do cell phones and shoes is because shoes act as a collector of microbes. As you walk around, you're collecting microbes on your feet. So the microbes that are on your shoe sort of give a sense of what microbes are in that area. The microbes on your phone give a sense of your microbes. So you shed your microbes onto your phone. And so by doing this at a number of events all around the country, we're asking what we call a biogeographical question, like what microbes tend to live in different areas of the country, et cetera. And so we can also hope to engage people in the process of doing science by having them sample their own cell phone and their own shoes. And that's what we're going to do. Everyone that comes to the Spurs game tomorrow, we have sampling kits, and not everyone in the audience, everyone who comes from the NSTA, we're gonna sample cell phones and shoes, and those can get sent off as part of this project. There's been some previous work on cell phones and shoes, and we'll be building on that with this study, and we'll also be able to compare our results to similar surfaces on the International Space Station. The last part of this is the microbial playoffs, and the idea here is from each venue. So tomorrow at the Spurs game, we will collect samples from the ball, from the court, from surfaces of that venue, and then bring them back to the UC Davis, and I will grow them up and choose the best one, the fastest growing microbe from that place, and then send that into space. So every venue will get to contribute one microbe that gets sent into space, and then the astronauts will grow those up and they'll compete against each other in space. It's mostly because it's cool, but there is also the question of some microbes are known to grow differently in microgravity environments and on Earth, and we'll do a duplicate of the experiment on Earth at the same time, and we'll expect to see differences in some microbes as to how they grow in space and on Earth, and we can look at that further.
Any chance this can replace the puppy bowl?
The what?
The puppy bowl.
We'll do what we can. We'll do what we can. So yeah, people will be able to root, and we're going to have a Super Bowl rematch with microbes from the Ravens and microbes from the 49ers, and there'll be lots of cheese around this aspect of the project, but it will also be awesome. I wanted to talk just real briefly about what the data from these experiments will look like. So if, let's say, someone did a project like this in their classroom or contributed samples or was involved, we want people to not just contribute samples and then never hear anything about it. We want to have a website where we show the data, the results of the experiment, where people can go and they can interact with the data and tell a story about what's going on, about the science. That's not as easy as it sounds. So let me just show some of the problems we have with the data. So what we're doing is we're sequencing the 16S gene of microbes, and all you need to know about that is it's like a barcode. So if we sequence this particular piece of DNA, we can say, oh, that one is E. coli, that one is Streptomyces. So we're going to sequence this little piece that allows us to identify what organisms are present. And so what we get is we get a long list of organisms that are present in a sample. If you take a sample off someone's cell phone, it's similar. There could be 100 different microbes on that cell phone, which is crazy to think about. And so you'll get a list of 100 microbes, and you'll also get a list of how their relative abundance. You know, there was a lot of this one and not so much of this one. So one way to visualize that data, this is from an actual phones and shoes experiment that was done previously. And on the left are phones and on the right are shoes. And each bar represents a single sample. So this is a single person's phone. And each little color on here is a different microbial species.
Oh, that's so cool.
And so you can see, again, there are a lot of different species of microbes on phones and shoes. But you also can see immediately that there's a difference between phones and shoes. You know, the species that are represented are different. In fact, we can look at a sample and we can say, oh, that came from a shoe or that came from a phone based on what microbes are present. So that's one way that we can represent the data. Another way is to cluster these samples. And I'm not going to talk about the math behind this because I don't understand it either. But basically, shoe samples here are shown in orange and cell phone samples are shown in blue. And the kind of things you can draw away from this representation are that shoes tend to look, they're close to each other. So my shoes will tend to look like your shoes will tend to look like your shoes. But cell phones tend to vary more. The microbes that people are shedding on their cell phones vary more. And you can also see that cell phones and shoes, again, are quite distinct. So these are the kind of representations of data that we hope to have for the samples we collect. That people will be able to go and they'll say, oh, you know, the samples at the Spurs game, you can see how they're different from other parts of the country. So how do we collect samples? Well, not like this. It's actually basically fancy Q-tips. I just want to real briefly show the sampling kits that we're using because any of you that come to the Spurs game tomorrow, hopefully all of you, will get to do this. And we have some of these that actually we can hand out and you can take back and do in a class. And the kit is real simple. It just contains a couple plastic bags, some sterile swabs, and a barcode. You put the labels on the bag, put one on the informed consent form, which everyone is required to sign who does this kind of thing because you're contributing your microbes on your cell phone, so you have to agree to that. This data is anonymized, so you won't know what samples came from which person, but there's still a consent to participate. Take a swab out, swab your phone. This is me, I'm the last person on earth with a dumb phone. Put it in a bag. Swab your shoe, put them both in a bag, and well, that's interesting. I have the address of something on there. That's an idiot.
That's this address.
That's this address.
Yeah,
I'm not quite sure how that happened. And then Darlene wants to talk again.
Yeah, thank you. Thanks for doing that. And we'll come back for some Q&A. Okay, so I promised that I would explain some of the other partners. This is SciStarter. This is the sister site of Science Cheerleader. So the Science Cheerleaders are living beings who do things. We draw most of our projects from SciStarter, which is a website that has something like 500 or 600 citizen science projects listed in there. So we aggregate them, curate them, and make it easy for the public to learn about them. In fact, we work with the National Science Teachers Association to be able to just serve up a project of the week that you might want to consider getting your classroom involved in. This is one type of project. They are in almost every single category. And you can also search for things, show me things I can do at school, in my school. Show me things I can do at the beach. Remind me, Cosmo Quest isn't in there yet, but we want to get it in there. She runs an amazing citizen science project. And again, the Science Cheerleaders, just coming back to that, we are moving from being just the promotional face to actually leading some research in this regard. This is a picture of Heidi who cheered for the Kansas City Chiefs. And I don't remember offhand what her STEM background is, but she led a cheer that ended up setting a new world record with Pop Warner football and cheerleading organization. And that was fun. They all cheered for science. Yay! Did you have a question, Russell, or were you just?
Oh, I was just going to say, Wendy is a co-principal investigator on this project.
Yeah, so Wendy, I mentioned her earlier. She's not here, but ironically, the game tomorrow is the Spurs versus the Kings. And Wendy, who is getting her PhD from UC Davis, one of the co-PIs, used to cheer for the Spurs, the Kings. In fact, that's where she is this weekend, right? Isn't she trying out?
She's trying out for the Oakland Raiders.
Oh, for the Raiders, okay. Yeah. So 250 members. These are some of the types of work that the science cheerleaders do. You can see citizen science plays in there. Every appearance they do, they tie to a local citizen science opportunity for teachers and the audience to get involved. So their job really is to playfully challenge stereotypes, inspire young women to consider STEM careers as well, and then let other people know that even if you're not a scientist, you might not be a teacher or policy maker, there are other ways that you can make a difference with science and connect to science. And we're putting together a Science of Cheerleading app now.